Okay, so while we figure out teams, let's just get started. So my first question, I'm gonna give a little background, right? So, ex vivo CAR T therapies, despite kind of showing really strong efficacy even up front, has generally been introduced in later lines of treatment. And after patients have kind of progressed through other options like chemotherapy or antibody based therapies. And so my first question is maybe what would in vivo CAR T have to show either clinically or economically to move into earlier lines of therapy? Do you think that it's going to follow a very similar path as ex vivo, kind of starting from the very bottom and then moving itself up? Ralph, maybe I can have you kick us off.
Sure. I think it will follow the same principle. Typically, what you need to do is, in early clinical trials, you use late lines, and those first approvals are late line patients. Those are very, very sick patients. And in order to move up, when you go to earlier lines, those patients are not nearly as sick. So the risk-benefit ratio changes. And so typically, you do need to show in a clinical trial, the structure of a clinical trial changes. It goes, a randomized control trial, not against a placebo or best supportive care, but it goes against standard of care, typically. The endpoints typically change. So you go from overall response and CRs, you go more to progression-free survival or event-free survival even. And I think for in vivo CAR Ts, it will have to follow that same principle.
And then I guess, Shon, for autoimmune and for allergies, do you think will differ?
I was reflecting while you were answering. I agree. I mean, it's going to be a very competitive space for going into indications where ex vivo CAR Ts already exist. It would be great if we could find novel indications that just bypass that, because if you're first, you have a huge advantage. It's the reality that after we're going to have probably Cabaletta and Kyverna are going to be the first with the CAR T in autoimmune indication, and they chose specific indications. And then any CAR T that wants to come into those indications will have to prove that it's better or safer in some way. I don't think being cheaper is a category that the FDA, I'm curious what you say, because coming in with an in vivo approach that's just cheaper, would that, please comment on that.
I agree with you. I think from a regulatory perspective, the cost economics don't matter. It has to be safer, or it has to be non-inferior with additional benefits. That's what you need to get. The economics, I think, influence investment into your company. So your ability to do that. But just that it's cheaper but no better is not going to help you with approval.
I can put some other consideration more on the cost effectiveness. Over here, we're talking about those kind of approaches. Money is not a consideration if the FDA would approve it or not. However, money is the consideration if your patient dosage per patient cost is going to limiting your company growth. We see a lot of novelty technology, but not being able to sell to the client because manufacturing cost is just relatively too high, especially consider gene therapy being a very novel approaches with AV scenarios. Like your development cost is so significant, but you're focusing on rare disease, your patient pool is very limited. How to being beneficial in the market with this singular drug, only aiming maybe 2,000 patients per year, it's going to be very challenging for designing your price and all that, right? However, with in vivo CAR T approaches, you're considering from a singular personal designed drug into more of an off-shelf options, and your dosage of the patients going to change directly from a drug product with a cell therapy and approaches directly into an off-shelf viral vector or mRNA. Those are changing the game. So overall, you don't have to per lot dosage manufacturing for those ex vivo programs anymore. However, I don't think that's going to affect the final cost by much. However, the overall turnaround time and the additional development in the cell therapy can be eliminated. That's probably the beneficial for the client consideration to move from ex vivo to in vivo in this standpoint.
There we go, I think. This may be more of a question than an answer, but the other opportunities we see are the opportunities to avoid lymphodepletion and the trials currently where we are unable to repeatedly dose patients with CAR T cells just due to the toxicity, and given the short lifetime of some of the in vivo CAR T, it may be much more feasible to dose on a schedule. So all of those things may help us, but how those will be evaluated going forward is a question we have as well.
I think overall you separate the regulatory aspects from the market aspects, and the big pharma companies that already invested in ex vivo CAR T are also investing in vivo CAR T, and they may decide that that is the appropriate business case for them, and eventually maybe discontinue ex vivo if in vivo shows sufficient efficacy and safety.
And I think lymphodepletion is a point because that is a safety consideration, and so that could very well be part of why in vivo CAR T would be safer and therefore affects that risk-benefit ratio.
And we definitely have patients that are not candidates for lymphodepletion as well, so it also opens up a slightly different patient pool.
Agree with that point. And building on that too, bigger patient pool just from a practical standpoint of there's still a relatively limited number of centers where you can effectively do ex vivo and have the infrastructure to do that well.
So second question is maybe going to create some more discussion. Is, we've seen kind of two dominant modalities emerge in vivo CAR T. You can split them up to TLMP and TLVV. To all of our speakers, if you were starting a company today, which would you choose, and which do you think has a clearer path to clinical and commercial success? I know it is, science modality dependent, so I'm going to broaden the question and include, which do you think currently has the better position when we consider access to technology, licensing, things like that, platform?
I'll start because I can start with the favorite consultant answer, which is it depends. and then I think I can hand it over to Scott because he already talked about this in his talk.
Yeah, I really do think it is application dependent. And so if I was starting another oncology-focused startup tomorrow, I would absolutely use a virus-based approach, an LVV-based approach, for the reasons that I mentioned. I think if you really want cancer treatments that are going to be durable, I think you need that stable integration. And without the invention of some other new technology, I just think the transient approaches such as LNP plus mRNA are just not going to get you there. That said, I think LNPs are really attractive for other applications where there's actually some big safety advantages to having that transient nature, and to let Shon and others speak to that.
I agree. If I was starting an oncology-directed startup, I would also choose integrating approach. So I do think that there's a big difference there, even though I've spoken to some folks developing CAR Ts that are transient mRNA LNP for solid tumors, actually, and they just believe that you can redose sufficient amount of times. Their argument was that CAR Ts sort of exhaust anyway and don't do well in the solid tumor environment and don't persist, which are all things that you're fixing. Hopefully your work is productive. Then maybe just a dose of fresh transient CAR Ts every few weeks would be better. So you can see these modalities going in every direction in the future. But right now, with the data that we have, I believe the transient CARs are better for indications where you expect an immune reset, for example, or just want a transient CAR. Like Capstan started out with a FAP CAR. Right? It's not what they ended up developing, but they wanted to work on cardiac fibrosis and didn't want a permanent CAR that would continue to eliminate fibroblasts in your body or prevent tissue repair. So there are use cases for each. But I do want to say something about COGS. I've been in the trenches of both. Interestingly, as technology develops, the COGS do go down, and they're going down kind of in both departments. It is quite expensive to make an LVV-based drug, but it is quite expensive to make a targeted LNP as well. If it's not targeted, you probably need to dose a lot more of it. All these things kind of balance out. So right now I view both applications costing about the same. eventually, maybe not right this minute.
I would echo to that construction. So both TLMP approaches, there's a lot of elements in those construction, including like we talked about antibody discoveries, as well as mRNA, including plasmid works, eventually into LNP formulas. Those all cost a lot. LVV, you have the same thing. You're not going to use the regular third generation packaging system with for plasmid. You have to have a novel design of the fusogen and the binder system as well, right? So those kind of development all cost monies. But my approach is like, I agree with Scott, it's basically depends on what modality you're working with. Let's say if you're working with a nail, a screwdriver is not going to do the work, right? So you always want to pick a hammer. So in the same case for this kind of modalities, what are you trying to fixing in oncology, or you're trying to fixing more of a surface issues? You select a different tool that's more feasible for the interactions. And later on, it will be more beneficial to have the correct development to guiding those order of development works as well. So from my perspective, the technology platform is essential, first of all, knowing that's effective to the modality you're working with. And moreover is when you're selecting those kind of approaches and to have more of a systematical building into a regulatory guideline, which for me, LVV has more safety concern at the moment, but it can resolve that with more proven track record. In the future, that might be more accessible. Well, in the short term, LNP for formulation has definitely had an easier approach and faster track record to be able to get into the market. Those are the different beneficial from each modalities. As a new startups, depends on what you're focusing in those kind of modalities, as well as those kind of technology you're based on is going to be your selections.
I still think that there's such a big space for innovation in the LNP, the mRNA LNP space, that there really is a lot of opportunity to either get away from antibody targeting and go to novel targeting moieties and or various innovative strategies for extending the lifetime of the efficacy. But also we know that in patients, a single modality is not going to cure cancer in most cases. So having the transient nature of the LNP also gives you the opportunity to really think about combination therapy in different ways. So it does seem like there's a lot of opportunity there to be creative and innovative.
Julie, I don't know if you can hear us, or can you comment and
What I could add is I think there's a misconception that LMPs can only deliver RNA species. And so there are a number of companies that are looking at delivery of DNA, which would extend duration of expression, delivery of multiple payloads in order to get the integration of the CAR into the T cell genome. And so I think to the point, there's still a lot of a lot more innovation that can happen on the LNP space. And so I'm on the LNP side of things. I do think vector has been very powerful, but I do see that LNPs, there's a lot more runway to go.
thank you for that. To our next question. Now kind of shifting to the therapeutic modality itself. I'd love to hear kind of what do you think is the single hardest unsolved problem facing in vivo CAR T therapy development today? Delivery, specificity, persistence, safety, or manufacturing, or other. And do you think that this can be solved by kind of improving existing technologies, or requiring to kind of taking a more fundamental new approaches? Just want to mention the audience, please feel free to participate. We're talking a lot, but if we'd love to get your insights as well. And Scott, maybe we can have you start for this one.
Happy to. I think I'll start with the answer that I think the most challenging single problem is the one that's facing all anti-cancer cell therapies, which is solid tumor efficacy, and doing that in a way that is safe enough. I think that is really the frontier that is hardest and has the most headroom to really, if these are going to be widespread drugs that have a chance to give patients really durable remission for a lot of cancers we can't treat yet. And I liked how, like, Julie, in your talk, you said like something along the lines of, the biology is still the ceiling. So I think that's a really important thing to consider. Everybody's excited about in vivo CAR T right now, as am I. But once we solve all these challenges around COGS and scalability and all these modality things, there's still going to be a huge amount of disease indications out there that we can't treat in that ceiling. Really, it really is the biology. We're still up against that same biology. And then I think, like, since that's a little bit of a cop-out answer to zoom in on the in vivo specific problems, I do think it's a nuanced answer, but I think the single hardest things is the trade-offs, right? Like, it's everything that you do is counterbalanced. So it's like, anything you increase with respect to potency or transduction efficiency, you increase the safety risk. And so I think, like, and also, like, improving the scalability and the COGS, there's a lot of trade-offs and a lot of things that are kind of, additional things added to compensate for something that adds complexity, adds safety risk, actually adds cost. And I think it's really getting the right balance and figuring out exactly where those thresholds are. That is a big challenge.
Yep. I can go next. So over here, I think Scott makes a very good point about the overall challenges, but I would like to discuss more on the current threshold that we are meeting day to day. So for in vivo CAR T, we all know, like, compared to the ex vivo CAR T, you have a very distinct targeting T cells that being enriched during the prior process before the cell therapy end. So you basically only working with the T cell transfection during the cell therapy and modalities. However, when you're talking about injecting virus, vector, or mRNA into your body, your systematic approaches consider that the environment changes compared to cell therapy manufacturing scenarios is totally different. Like, each individual body of the patient is different. Like, although it's not unique as what ex vivo programs is going to be meeting with, but in internal body, you're not only facing T cell in this case, is how to specifically targeting the T cells or the targeting cells that you're looking for with the in vivo CAR T approach is very difficult for us to be able to approach this, including fusogens as well as binders specificities to eliminate any off-target capability as well. So with that, ProBio has looked into some of the technology trying to resolve those issues. On the approach over here, one of the things we're seeing is when you're injecting LVV, for example, into patient bodies, you see off-target issue happening. Sometimes they're targeting, making CAR B cells, and the CAR T and CAR B cells trying to kill each other. Those kind of things are majorly caused by surface protein expression from your manufacturing process of the LVV. So having those kind of novel technology, which ProBio helps to develop a very good backbone to be able to eliminate those kind of things from happening from the beginning, mutating those protein expression during the manufacturing of LVVs. So those kind of programs to be able to eliminate those kind of challenging points, but we still wanted to see more better methodologies or novel technology to be able to have specificity targeting on the in vivo CAR T approaches to be more safe, like Scott saying, is the end goal to be able to dose the patient without any risks.
I don't think I have much to add. This is a very comprehensive discussion, I would say, but in general, on the CMC side is, if it's been characterized by some, I think a former FDA commissioner, that when you look at the BLA in cell and gene therapy, the focus of the review is on the CMC side, and really most of the effort goes into that. And even though an LNP might look simpler than an ex vivo genetically modified cell therapy, it's still cell therapy with all the complexities. And we've heard from the presentation from Julie too, is how complex those LNPs are. And so I think generally speaking, CMC is complex, but truly speaking, it's the trade-offs you need to get for if you need a high dose, the specificity, the potency, etc. Those trade-offs are really going to be the true challenge.
Yeah, I agree that the discussion is great. And the only thing I can think to add is I think that there's the opportunity for multiple layers of innovation, and combining those layers of innovation in a package and being able to move a multiply innovative particle forward quickly is maybe the challenge that I see that is somewhat unique.
Julie, do you want to go next?
Um, I I don't think ex vivo is going away, and so there is a large potential for in vivo CAR T to take off. Um, it's but there's space for every, I think, modality depending on indication, on what you're actually trying to do. Uh, yeah, I guess I think it's gonna cover.
Do you think one of the challenges is ex vivo, especially with kind of automation coming in, that and that you have to, your in vivo now has a different risk profile, and that you're coming in, you have to prove that you're safer and better than ex vivo, while other companies are trying to drive down the cost of manufacturing for ex vivo?
I see ex vivo being very different than an in vivo CAR T. There's a lot of there's just so much you can do when you culture cells outside of the body, that you can't do directly in the body. And every patient will be different. And so I, coming from a CMC perspective, how do you control the response? In an ex vivo setting, it's just more something that you can automate. Um, there's been a lot of work towards figuring out what the right T cells are for specific indications. How does the patient starting material influence the drug product? And so all of that will still need to be found out for in vivo CAR T approaches.
Maybe just kind of to recap, because when you sent the questions in advance, and I thought all of those things are the most challenging. And maybe to distill it to safety, efficacy, and complexity. There's so many nuances that need to be addressed. I think right now, in vivo CAR T engineering has to prove itself in terms of safety and efficacy in order for additional investment to be there for everything else to actually, like the challenges of commercialization or access, those are not today's problems. And today's problems, there's some toxicity associated with in vivo viral modalities and mRNA LNPs. And there's efficacy is looking okay, but maybe not great. So those are the biggest challenges right now, is can we actually compete, but also like show value for this modality? I'm sure we will, but that's the current challenge.
I see a lot of parallels, having worked on allogeneic CAR T approaches after working on autologous, in that the allogeneic field has had to deal with similar challenges that the in vivo field is facing right now. So I think
the
one
allogeneic field didn't do very well.
yes. So you thought there would be a huge COGS advantage for allogeneic, but I think the more bells and whistles that you start throwing in, the more costly things tend to get. So for in vivo, that may also become an issue if we decide to stick in this target ligand and that other piece.
To the point you just made is it's amazing once you have a commercial drug is how well these companies improve their manufacturing. The autologous CAR Ts early on, lots of manufacturing failures, very difficult to get to enough manufacture for enough patients. They've substantially improved. Yes, they're still not, there's some that are blockbusters, but they have worked really, really hard to get their efficiency up, their success rate up. They've moved into earlier lines. It's really hard to get to that now. So, similarly with in vivo CAR Ts, is you have that bar, and it's not, that bar is moving. The autologous CAR Ts, they're going to move that bar further and further. So it's not like today's challenge is this bar. No, by the time you're in the clinic, you get the data that bar has moved.
Yeah, very great discussion. I have a question regarding to, like Ralph mentioned earlier, for autologous, it's still, the efficacy depends on the patient, it's self T cell. So in vivo, when you think of the concept, it doesn't necessarily change the patient's constitution, right? So it depends on how do you improve patient's T cell when you treating them in vivo, doing it.
I can just cover some of the understanding of that, not really a solution over here. So the differences over here we're talking about for in vivo CAR T approaches, you're making a product. If it makes sense, it's an off-shelf product that LVV or mRNA approaches, you're making this drug product at the end of the process. As compared to an ex vivo programs, you're making a cell. The cell consideration is way harder to control ex vivo programs in this case, especially patient-to-patient difference controllable, because basically you can demand the patient cell to grow exactly the same performance as the other one. However, I do have more control over CMC strategy, over antiviral factor production, even more with the mRNA approaches. So those control is stringent at the CDMO side, can be well controlled. However, the only thing I cannot control for in vivo CAR T is the patient status. To be honest, our patient differences, not only by patient differences, also day-to-day variation as well. Today I'm healthy, a lot of activated T cells in my bodies. Tomorrow they cannot be silenced out. So those are the differences we cannot predict, nor can we either control in those kind of changing environment. We have to admitting to knowing in vivo CAR T, you're gonna meeting those scenarios from the day-to-day cases, differences. However, what we focusing on control the strategy is more on the CMC approaches to making sure your product doesn't have variations, batch to batch, including product to product.
Good points, and I'd just like to chime in and answer that from an efficacy perspective, and Julie made some nice points to this and others, I think, earlier as well, where, if you look at the auto, like CD19 programs, there's a lot of data coming out now, because it's been in the clinic long enough that yes, the patient-to-patient variability, there's definitely profiles of the T cells that correlate with success or not. And that is something that if you just have a generic Gen 2 CAR, you can't control in vivo. And so it's something that definitely needs to be addressed. I'll just speak for how we're approaching it at Outpace, which is we're really trying to solve that problem by coming up with the right efficacy technology that can reprogram the CAR T cells so that even if there is a predisposition for a patient's T cells that aren't going to be as good as a different patient's, we can correct for that enough. So it's a big challenge, but I think we can do it. That's why we're doing it. That we can have efficacy technology that reprograms the T cells so that you still get the right phenotype, the right functional persistence, et cetera, But without stuff like that, I think it is going to be really hard to get consistent efficacy with an in vivo delivered product for the reasons that have been mentioned.
Except for in allergy patients, because they're healthy. Anyway.
Yes, no, exactly.
We can be clever about where we point
Yes.
our technologies at this point.
100%. My coming purely from like an oncology perspective with my answer.
No, of course, I'm just adding some humor to the point. But your point is very well taken. A lot of people are worried about that heavily pretreated cancer patients without additional editing or bells and whistles added to your delivery modality are not going to have good enough CAR T cells, because taking the CAR T cells out of the body, the T cells, to engineer them does give you some control. With cytokines, with activation, you're putting back a product that's a little bit more expanded and activated than what a patient might do. So yeah, it will require work like yours and others that are already thinking in that direction to further modify the in vivo product, not just a second generation product.
Definitely.
Organizers for putting this symposium together, and all speakers for sharing your expertise. My question is related to one of the topic we discussed earlier, between the comparison between LVV and mRNA LNP. So specifically, it will be focused on the interaction and conversation with the different regulatory agencies around the world. Because earlier, one of the point mentioned is that the theoretical safety risk is much higher with LVV because of integrating nature of the virus. Can you share a little bit more about your interaction with the agencies, maybe your companies or your colleagues, or maybe your clients' conversation with agencies about their input on this specific topic on LVV? Thank you.
I can share some perspective from past experience of mine with regulatory agencies with an LVV in vivo product. It goes again to the risk-benefit ratio and the target indication. So if you're going into cancer patients that have, to the unmet need population that has exhausted other options, they're willing to tolerate some risk. And we don't know what the risk is of off-target transduction yet. A lot of the LVVs that are being engineered are very sophisticated and eliminate off-target binding greatly. And then there's empirical ways to measure biodistribution and estimate how much off-target transduction you may get in the body. So you reduce the risk and you do a risk assessment, and it's accepted in oncology patients. Probably not accepted in autoimmune patients or allergy patients at this time.
I can add additional information based on the communication with some of the couple clients. So in this kind of off-target issues, we do see a major challenge because LVV had the potential. However, has maybe in my ignorance, have not been testified it actually happened. So we only know the risk if it happens, what it's going to be. However, nobody actually to demonstrate those kind of off-target issues to be demonstrated, a failure mode or what kind of issue it's going to cause if it does happen. So right now, FDA is potentially just trying to making sure you can demonstrate the risk mitigation plan like we previously said. It doesn't happen and how you can control those kind of risks potentially if it happens. However, the risk is more than likely we don't know. So how to prove something never happens, that is a very challenging topic in this moment. So FDA is more tolerable saying like, okay, as long as you have a consideration knowing this risk exists, and trying to avoid every possible things you can do for that to happen again, internally. So that's the requirement.
Thank you, everyone. This has been very insightful.
We can hear you.
To what extent do we expect in vivo CAR Ts to address the main bottlenecks that are faced by ex vivo CAR Ts, in, particularly in solid tumors, because we were just talking, you guys just raised that, specifically in terms of like TME suppression, antigen heterogeneity, T-cell exhaustion, poor metabolic fitness of the T-cells, even if they do manage to get home to the tumor site. So I guess I'm just curious, because I've heard someone address this challenge as like holding the hammer and going around looking for your nail, because if you're trying to implement the same technology that we have for ex vivo CAR Ts to in vivo CAR T, does it really make sense if we're using our existing technologies for non-solid tumors in solid tumors? How does that really ease, or how does that answer any of the questions we have?
Good question. I can start. And yeah, I think it really comes down to, this is kind of my hot take. I think if there's a challenge that ex vivo, from an efficacy standpoint, if there's a challenge that ex vivo CAR T can't do, I don't think in vivo is going to be able to do it. And the flip side of that is I think if there is a technology, whether that's counter-exhaustion, cytokine support, things like that, if it works in ex vivo, there's paths to put that into in vivo. So I think it's a really good question, but I think that the oversimplified answer is that for those types of things, the challenges are the same. And the technologies that are needed for both in order to go after solid tumor efficacy. I think like where there's maybe some opportunities, I saw you raising the microphone, because you had some really good points earlier about the opportunity for combination therapies. And also like Shon, you mentioned like, the possibility to like people are doing like the possibility of redosing when you have some of these new modalities. And so I think like there's certainly opportunity to get creative, and you could imagine doing combinations of things in a more creative way than you could do with ex vivo. But the fundamental problems that we need to solve, they're the same.
Somewhat more optimistic, just because I've watched the journey of radioligand therapy, which is where we inject first an imaging isotope. We look to see whether or not we've got the specific receptor upregulated, and then we use a therapeutic isotope. And that typically now goes through 6 cycles, 6 weeks apart, with, in many patients, tremendous reduction in tumor burden and huge shifts in the tumor microenvironment. But those patients are still not cured. And so, there is a need to follow that or to combine that with other, and certainly with some sort of an immunotherapeutic, I think, to actually achieve a longer lasting response. But watching that and watching it move from a late line therapy to now an earlier line therapy has made me much more optimistic that that's possible, and that the combinations that are feasible will help to cross some of the barriers, especially the tumor microenvironment barrier. And also, I think being able to give cytokines and to change T cell phenotype is certainly something that we can do, and is another possible addition that could address some of the challenges.
Since it's already in the solid tumor topic, I didn't mean to off topic, but there's another modality. It's called a CAR-NK, and it seemed to have less GVHD and better tumor infiltration. So in kind of this modality and then the current in vivo or the ex vivo CAR T, how do you feel about the potential for the CAR-NK field?
Good question. I can start there, and this is very much, I think, my personal biased opinion. I think because yes, people are doing CAR-NK, people are doing CAR macrophage. I think it's kind of died down, but for a while it was a really hot field. People trying to do other types of engineered cell therapies, not just T cells. My personal bias is that I still think T cells are the way to go. I think they're the proven modality. I think there's a lot that can be done with additional technologies that recruit the endogenous NK cells and potentially endogenous macrophages, like repolarizing the endogenous macrophages. I think there's enormous potential there. But I think, going just after a different cell type is not going to solve the fundamental problems. There's still exhaustion is still a problem for CAR-NK and solid tumor, and NKs have some unique things about them that kind of create a different set of problems that need to be solved in order to get solid tumor efficacy. So I think there's potential there, but that's my hot take, is just switching cell type is not going to do it. And I think there's enormous potential through technologies or combination therapies to recruit the endogenous types of those cells to the fight in addition to the engineered T cells.
I just wanted to quickly jump in. It seems like there's real potential using in vivo modalities to strike multiple or address multiple different immune cells at the same time. That could be really transformative, because you wouldn't want to manufacture a therapy with multiple different kinds of immune cells, but with the right kind of synthetic biology, for example, ligands that address different cell types, or promoters that can selectively activate and program these cells in different ways. That seems like a space where in vivo therapeutics could really distinguish themselves. So I just wanted to add that, since we were talking about the CAR-NK.
It is happening. When you use CD8 targeting, you get NK cells as well. And the ligand that we're planning to use also targets NK cells. So it could be part of the package of in vivo delivery. It seems that clinically, people don't track NK cells as much as they do T cells. And maybe that's because they don't expand a lot, and that's only in the LVV modalities. I don't know what else to say about them. NKs are very exciting, but they do have some issues too. I don't think they would solve the problem alone.
Yeah, very much agree, and thank you for the question. I mean, it's a really fun space to dream about, and I think it's also a liability too, right? I think that is something that is underappreciated in some of the field with in vivo delivery, is that most of these approaches, there is multiple different types of immune cells getting transduced with virus, and that's both a potential opportunity but also a liability.
Sort of specifically to the NKs, the in vivo, even if you target NKs in vivo, they don't expand And so if you want to efficiently use the NKs, targeting them directly with an LNP or so is probably not as effective because you would need to use a lot of material to target them. And so the indirect effect of recruiting the NKs might be the more effective way to include them in the response.
Very much agree, and very good point. And that's why you see people doing like the membrane tethered IL-15 or like the trike with the IL-15 whenever they go after the NK.
Okay, we're kind of up on time. So last question, as we're kind of wrapping this up and looking ahead, what does the future look like? Are there necessary capabilities or features that's really kind of essential for the success of an in vivo CAR T therapy? And also in terms of technologies, capabilities, is everything already in place to enable, successful commercialization of an in vivo CAR T therapy?
I can start off. As a CDMO companies, we're ready. We're very excited about all the novel modalities right now, talking about in this forum as well as future modalities that our clients being able to bring it to us. So we're excited about it. And the platform processes right now, we're trying to do one thing is very essential to a CDMO company is feasibility. So we want to making sure every client bringing those kind of new modality, new construction to us, our platform processes can help them accelerate the programs. So without much more, like I said previously, process development necessaries before going to a tryout of an IND approaches. So with those kind of accelerated programs, so we can realize those kind of dreams way ahead of their schedules. So potentially to save a lot of cost as well as timeline. So bringing those new modalities to the patients as soon as possible.
I might have a slightly less optimistic perspective, but yeah, this year is, I think, going to be interesting, right? Last year there was almost nothing in clinical trials. Now we have over 20 programs globally in clinical trials. So the data isn't here yet to tell us what's going to happen. And so I read two different articles on the EsoBio data, and one was super enthusiastic, and the other one was, oh, did you look at those adverse events? And I think so. It really, there's a lot of data that needs to come out. I think in the presentation of Julie, you've also seen how, that on the CMC side, there's a lot that needs to be done still on the manufacturing side. It feels a little bit like the process of the product from 30 years back from monoclonal antibodies, is that we don't necessarily have the right methods developed yet. There is impurities we need to be concerned about. That then goes to the raw materials you're using, etc. So I think there's a lot of work that needs to be done, but critically is this initial set of safety or clinical data is really going to drive what's going to come next.
I guess I see several opportunities for improvements, and I do think improvements are probably needed for in vivo mRNA CAR T, but there's a number of options to make them more efficacious or to make them last longer, and or both. And redosing, getting a better sense of what the cost and the efficiency of redosing. There's so many opportunities, but yeah, I do think there's innovation needed, but there are a lot of opportunities for that innovation.
Thank you. And Julie?
Yeah, I'm having gone through the autologous ex vivo path, and then allo had this initial excitement. I'm very curious to see what the clinical data end up looking like this year, as I think everyone's excited about in vivo CAR T, that's for sure. But it's LVV, LNPs, even though there's been advancements, there's still a lot more that we don't know. And so, I guess we'll just see how things go this year.
Yeah, cautious optimism.
Yeah.
I'm very excited about this field, and I've been in vivo CAR T has been my passion for a long time. But if you speak, if I speak to investors, any investors, 100% this is the future and it's going to work. But if you look under the hood and you're a scientist and you're really looking into it, there's a lot of things we don't know and a lot of complexities, especially in the manufacturing. And it's not it's not done. It's not a done story, and there's a lot of investment in it though. So maybe in the next decade, we'll see a revolution where in vivo CAR T is really taken over, and we know how to use it. But it's still a risky business relative to things that we know work.
Absolutely. I want largely agree. I think also singing the tune of cautious optimism. I guess the one thing I would add is, back to your question, I think we've got all of the building blocks that we need to do this, but we've got a long way to go. And it's truly an exciting time. It's going to be, the next couple of years are going to be really informative and really exciting.
Thank you.
I wanted to add in last point to correct the false promisings over here. Now, I'm not saying in vivo CAR T is going to be easy to overcome with a lot of the challenge we previously discussed, right? So being optimistic is knowing like those kind of new modality and new approaches to fix the disease we won't be able to overcome in the previous track record. And having new tools like we previously have hammers, we have screwdrivers, now we have saws. So knowing those kind of things are getting me excited. But we know like using a new tool and fixing the new problems is going to be always challenging. But to be honest, like ProBio as a CDMO, we're always going to be there to support our clients as well as to bringing some of the technology we develop as well, together with the innovations all the clients are looking forward to have, to together walking those kind of hard way to pass to reach out to success.
Yeah, thank you. And in conclusion, I'd like to thank our wonderful speakers for all volunteering your time. Thanks to everyone in the room here for taking the time to participate. And I hope the discussion doesn't just stop in this room, that we continue talking. Sounds like there's still a lot to work on, and I think always collaboration, discussion really kind of moves things forward. Finally, I think we have drinks and snacks. Please join us for some happy hour. Keep talking. We can chat about